Namaskar, sorry for a little delay.
Good morning, sir.
Good morning.
Hello. Hello. Good morning and namaste, everyone. We welcome you on second day of OCELS 2021. I hope you have enjoyed day one. Second day, we'll begin with Dr. Sudhir Verma, sir, who is Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Mumbai. He has profound knowledge and very multitasking personality. He involved in teaching, research, and science communication. He is a certified trainer in research-based pedagogy by Shepherd Hallam University, UK. He has developed various e-chapters, video for EPG Pathshala, Swayam, Prabha DHT channels for undergraduate and postgraduate student in the subject of immunology and evolution biology. Besides his research in epigenetics and immunology, he is also involved in STEM education research. He is bestowed with various fellowships and scholarships. I have avoided to include in this particular slide because the number is very high. Okay, we have to get knowledge from Sudhir sir. So I've cut short the introduction. He has been the judging panelist for Global Undergraduate Awards 2019-2020 for Atal Tinkering Lab exhibition. He was a resource person for NCRT, NRCB, NIPA, Delhi. Dr. Verma is a certified innovation ambassador for Ministry of Education and currently is also working as fellow at Institute of Lifelong Learning, University of Delhi. As far as his research and publication goes, he has wide number of publication chapters in book, international publications with reputed journal. I here welcome Dr. Sudhir Verma sir, and I am not coming in between you and Verma sir, as we have to get knowledge from Dr. Sudhir Verma. I welcome Verma sir, and I expect him to take the charge of the session. Thank you, sir.
Thank you so much, Dr. Mukesh, for this nice introduction. And a very good morning and warm greetings to all our participants of OCELS-21. Since we have limited time and we have a lot to discuss, let me, without any further delay, start sharing my screen. I'll just put off my video and start sharing the screen. Just give me a second. Dr. Mukesh, can you just confirm whether it is visible to you all or not?
It is visible, sir. It is visible.
Thank you so much. All right. So, as I expect, and I'm sure that you all must have gone through the schedule which has already been shared with you all on the Google Groups, wherein I am given a task to discuss about evolution and behavior with you in next 1 hour or so. Now, how I have planned this is we have taken a few selected questions or the problems which have been asked in previous years in various competitive examinations, particularly the CSIR-JRF exam. And these questions are based on some key concepts on evolution and behavior. So the idea here is to take these questions, discuss with you so that we can understand or have a clear conceptual understanding about the key concepts. And we do understand the logical reasoning behind avoiding the distractors and selecting the right option from those multiple choice questions. Now, before I start discussing those questions, here we can see the syllabus, which is there for evolution and behavior particularly. The syllabus is divided into 6 major subhead, as you can read here. So I'll just spend a minute or so to discuss with you what all is there, and then I'll move on to the questions. The first unit is on emergence of evolutionary thoughts, wherein various theories and the concepts about introduction of evolution, how is it different from creationism, what are the various theories which have been put forward to explain evolution, etc., has been included. In the second part, that is origin of cells and unicellular evolution, we do have the basic biological molecule origin, the concept given by Operin and Haldane, various experimental evidence with respect to that, origin of eukaryotic cells, etc. In the third part, that is on paleontology and evolutionary history, we do have the geological timescale and the evolution of primates, including humans. Further, in molecular evolution, we are supposed to cover the concept of neutral evolution, molecular divergence, and molecular clocks, and various key concepts of gene duplication, gene divergence, etc. Further, in the part which is there as mechanisms, we do have population genetics, particularly, wherein we are supposed to cover the Hardy-Weinberg equilibrium, convergent evolution, sexual selection, coevolution, etc. Lastly, in the unit which is on brain behavior and evolution, we are supposed to cover the key behavioral aspects which are playing an important role in evolution, that is skin selection, altruism, biological clocks, etc. So this is a brief idea about the evolution or the part which we are supposed to cover for this particular exam. Now, let's straight away jump to the questions and try to understand the basic idea or the basic concept behind each question and how we can conclude or reach to the correct option out of those multiple choice questions. So here we have the first question. Let me just read out for you and then I'll start discussing it. It says, in a population at Hardy-Weinberg equilibrium, the genotype frequencies are so and so. What are the frequencies of the two alleles at this locus? Now, this question was asked in June 2015 in part C. I would like to mention here that most of my questions I have taken from part C because those are the ones which do demand the logical understanding, clear conceptual understanding, and critical thinking. So most of the questions are from part C, though I do have included some of the questions from part B as well, which we'll take later on. So in this question, you can clearly understand that this question is from the population genetics part or the mechanisms part of evolution, which we just discussed a few minutes back. And it says that the population is Hardy-Weinberg equilibrium following population. So you must have an idea about what Hardy-Weinberg equilibrium or law or principle.
Sudhir sir, sorry for
Yes.
interruption. The pointer
Please.
is making noise.
Okay, so I'll avoid the pointer.
Okay, thank you.
Yeah. So this is Hardy-Weinberg equilibrium. You must have read in your undergraduate classes any large population which is randomly mating and which is devoid of any evolutionary force, that is natural selection, mutation, migration, or the genetic drift, does not show any change in the allele or genotypic frequencies, and hence it does not show evolution. So the key concept here is one should be able to calculate allelic frequencies and genotypic frequencies for a population in order to understand whether the population is following Hardy-Weinberg equilibrium or not. So here in this question, the demand is that the student should be able to calculate allelic frequencies. Now, there are two ways in which allelic frequencies can be calculated. First is through the number of individuals of genotypes. In this question, though, you can see the frequency of different genotypes has been provided. That is the another way of calculating allelic frequencies when the frequency of genotypes has been provided. But the same question can be asked in other way where they can give you the number of individuals which are possessing a particular genotype, and they may ask, what is the allelic frequency? So we'll understand both the ways and then we'll solve this problem. So if they have given the number of genotypes, that means they have given the number of individuals which are possessing a particular genotype instead of providing the frequency of that genotype. You can simply use this formula. For example, if A1 and A2 are two different alleles at this particular locus, and if they represent their frequency as P and Q respectively, their allelic frequency can be calculated by multiplying the number of homozygotes with 2 plus the number of heterozygotes divided by total number of individuals. Now the question is, why we are multiplying it with 2? We are multiplying the number of homozygotes with 2 because each homozygote is having 2 copies of that allele. And heterozygotes, since they have single copy of that allele, we are not multiplying it with 2. Whereas at the bottom, if you see, it's twice the number of total individuals because each individual again possesses 2 alleles and we are dividing it with total number of alleles. Hence, we have taken 2n. This is the point where students usually do mistake, and the distractors, the 4 options which have been given in the question, one of the option is intentionally designed so that students who do not multiply it with 2 at any of these places, they get the wrong answer, and that is how they get distracted. So this is one method of calculating allelic frequency. The other method is the one when instead of providing the number of individuals, they provide you the frequency of genotypes. As you can see in this particular question, they have given the frequency of 3 different genotypes. That is 2 different homozygotes and the heterozygotes. Now here, you can use these formula to calculate their allelic frequencies. You take the frequency of homozygote and add to it half the frequency of heterozygote for a particular allelic frequency calculation. Now, if I use this formula for calculation of allelic frequencies using the data which has been given in this particular question, we do get that frequency of A1 allele comes out to be 0.67. And how it comes? We have taken the homozygote frequency, that is 0.59 for A1, and we have added half of the heterozygotic frequency, that is 0.16. So it comes out to be 0.59 plus 0.08, that is 0.67. The other allelic frequencies you can either calculate using this formula, or since they have already mentioned in the question that this is the Hardy-Weinberg equilibrium following population, you can simply do 1 minus P in order to get the frequency of other allele, that is Q, because the sum of the frequencies of both the allele is 1 always. So in this question, the correct answer comes out to be 0.67 for A1 and 0.33 for A2, that is option 3. Now, this kind of questions are generally being asked as you see the previous year's paper. Moving ahead, the second type of concept. If you read out this question, it says the following diagrams represent various ways in which a character may evolve. So they are basically showing the pattern of evolution of a particular character, wherein each solid circle, filled circle, is representing a derived character, that means a newly acquired character, and the hollow circles, empty circles, they are representing ancestral traits. And they are asking which of the following is the correct definition for the character evolution patterns shown above in the figure represented by A, B, and C. They have given 3 different names under the 4 options: autapomorphy, synapomorphy, and homoplasy. In order to understand this question and answer it correctly, we must understand what do we mean by these terms and how we can evaluate the evolution or pattern of evolution in these characters. So for that, let me just remind you the concept of homologous and analogous organs, or the concept of common ancestry, which we have read in terms of evidence of evolution. Apomorphy is the term that we use for derived characters that sets the clade apart from other clades. Now, each line here, as you can see, they are representing a particular clade, right? And if you see in this diagram, which is shown as apomorphy, those two taxons, which have been shown with the solid circles, they are having a different character. And that is why they have got adjusted in a different clade. So they do have a derived character that is keeping or making these clade apart from the other clades. Similar term is simpliciomorphy, in which an ancestral trait shared by two or more taxa. So you can see in the figure, the term simpliciomorphy is depicted, wherein two empty circles that is showing ancestral trait that have been evolved as ancestral traits only from their ancestors. That means there is no derived character, there is no additional character. Now, based on this, let's come to these three terms which have been asked in the question. First term is synapomorphy. The word syn refers to same or shared. So this is nothing but another term which you all have used in your undergraduate classes. The term is homology or homologous organs, which depict the divergent evolution. So any derived trait that is shared by two or more taxa of shared ancestry or the common ancestry, they are supposed to be called as synapomorphy. So as you can see here, three filled circles or solid circles. The two terminal ones are basically the two taxon, and the below one shows their common ancestry. So any two taxon which do have a derived character from their common ancestor, together they are called as synapomorphy. In contrast, we do have another term which we call as homoplasy. In homoplasy, a trait gained or lost independently in separate lineages during evolution is referred. This is nothing but another term for your convergent evolution or the phenomena of analogy. As you can see here, the two solid circles are placed clades apart. One is in another taxon, one is in another taxon, and they have independently evolved this character. They do not have any common ancestor, as you can see here. This kind of evolution is called as homoplasy. And the last term that has been used is autapomorphy, in which a distinctive derived trait that is unique to a given taxon or group has been shown. So as you can see here, there is only one taxon which is showing this derived trait. And hence, this is the distinctive trait that is making this particular taxon unique among all other taxon. So this kind of evolution is called as autapomorphy. Now going back to the image which has been shown in the question, if you see A, there are two taxon with a common ancestor which are showing derived character. Hence, this is nothing but your homologous evolution or synapomorphy. If you see figure B, there are two different taxons which have independently derived a trait and there is no common ancestor. And hence, they do represent convergent evolution or analogy or nothing but the homoplasy. Third one, that is the C, if you focus upon, there is one taxon which has independently derived this character and which is unique. Hence, it is called as autapomorphy. So if you can understand this, you can easily reach to the answer, which is option 4 here, that A is showing synapomorphy, B is showing homoplasy, and C is showing autapomorphy. So if you clearly having an idea about how the evolutionary patterns are governed, you can easily arrive to the right conclusion. All right, moving ahead, this is the third question that I have taken. Now, this question has the key concepts from two things. They first want to check your logical understanding or critical thinking. And secondly, they do want to understand your understanding about phylogenetic trees. Let me just read out that question first. As a biologist, you want to classify three taxa, A, B, and C. So there are three different taxons, A, B, and C, and we want to classify them. You have information on three traits, P, Q, and R. So three different traits are there and three different taxons are there. The trait that is ancestral is counted 0, and the trait that is derived is counted as 1. So the trait which is not evolved, which remains as such like in ancestral forms, they have been represented as 0, and the traits which have been evolved, which have been derived, they have been represented as 1 here. The distribution of traits found in three different taxa are represented here in this table. So A, B, C are three different taxa, P, Q are three different traits. One represents derived character, that means a change, a new character is there, and 0 represents a different scenario where the ancestral form is going on. Based on the above table, the following cladograms were shown. Now, as you can see, A, B, C has been shown as three different cladograms here. And these lines which you see marked as P, Q, R, etc., they represent the, the trait which have been derived. And the question here that they have framed is, based on the trait distribution and principle of parsimony, select the correct option. And 4 options are there. This question was there in December 2016. And as I said, in order to answer this, you need to apply 2 things. First, you need to analyze this data correctly. And second thing, you must have the basic understanding of phylogenetic trees. Particularly, they are asking about parsimony method. So let's try to solve this question. Remember, they have said 2 things based on trait distribution and based on the parsimony principle. So let's try to analyze this question based on the trait distribution first. Now, if you see the A type of cladogram here, and you try to correlate it with the table of traits which have been given. In A type of cladogram, A and B type of taxons, they do have P and Q type of traits which have been shown by 2 different lines here. This one I'm saying. And then they are showing that B and C clades, they do have R type of trait as well. Now you can easily correlate from this table. P is there in A and B. Q is there in A and B, whereas R is there only in B and C. And you can correlate this table with the type of cladogram shown here in A. Coming to the B type of cladogram, again, if you see A taxon clad, they have shown the lines P and Q. That means they do have trait P and Q. If you see the B type of taxon, they also show P and Q lines. That means they do have traits P and Q. If you see the common ancestry of B and C, the R line is there. That means B and C both have the trait R. Hence, B is also consistent with this trait distribution data. Coming to the C, if you see A clad here, A taxon here in the C cladogram, there is only a P line there. There is no Q line there in the A taxon here, that is here. Means if you compare it or correlate it with the data of trait distribution, the P trait should be there in both A and B, whereas the cladogram which has been drawn in C, A taxon does not show the Q trait. Means on the basis of trait distribution, that means on the basis of this table, we can say that A type of cladogram is consistent with the trait distribution data. B is also consistent with this data, whereas C is not. And hence, if you just consider the trait distribution data, out of the 4 options, you can easily neglect the option 3. That means only C cladogram is possible. No, C cladogram is not possible because we can eliminate it based on the trait distribution. Now we are left with 3 different options. Either B is possible, or A is possible, or both A and B is possible. So based on the trait distribution, both A and B are possible. But remember, in the question they have asked, you need to consider both the things, trait distribution as well as the parsimony principle. So based on trait distribution so far we have discussed, let's now move on to the parsimony principle. You might have read how we construct the phylogenetic trees using bioinformatics tool. There are 3 different methods, particularly which we use. One is called as neighbor joining, another one is called as maximum likelihood, and the third one is called as and hence the parsimony principle, if you see, they say the simplest explanation with smallest number of changes. Now, so far we have reached to this data where A and B were consistent with the trait distribution, whereas C was inconsistent. What parsimony principle says is, in order to reach to the final stage, in order to reach to the cladogram which we can see now, the smallest number of changes must have taken place to reach that conclusion. Now, if you see the derived characters which have been shown here, if you see these cladogram which is shown in A, and you count the number of changes that it has undergone, that means P, Q, R, and R lines which have been drawn here, these lines I'm talking about. There are 4 such changes represented by 4 different lines, right? If you see the B cladogram and you count the number of lines here, there are 5 different lines which have been shown here. That means it has undergone 5 different changes in order to reach this conclusion. If you count C, in any case, though we have already crossed it, if we have already neglected it based on the trait distribution pattern, though it has 4 changes, 4 number of changes, we can simply avoid it because it has failed on the trait distribution data. So now we have 2 options. A is showing 4 different changes, B is showing 5 different changes, and as I said, based on the parsimony principle, we need to select the one which has taken minimal number of changes to reach to the conclusion. So according to this principle, if we go out of A and B, we can see A has taken 4 changes to reach this conclusion and B has taken 5 changes. So we can easily see out this 4 options, the correct option seems to be option 4. That means only A cladogram is possible considering both the factors, that is trait distribution and the parsimony principle. I hope this thing is clear to you all. As I said, this question tests 2 things. One, your analysis of the data which has been given, and second thing, the basic understanding of phylogenetic tree construction principle, particularly the parsimony method which you have seen here. So this kind of questions are generally being asked in CSIR net particularly. Now, let's move ahead to another interesting question, which is from the last unit, that is behavioral part particularly, though it is a mathematical problem, but it do involve the clear understanding of the concept of altruism, fitness, etc. Let me just read out the question. Inclusive fitness of an animal can be measured as a sum of direct fitness and indirect fitness. Imagine you have 10 offsprings. Through diligent parental care, 5 survive to reproduce. You give your life in a heroic deed to save a total of 5 of your nieces and nephews. What is your inclusive fitness? And this question was asked in June 2018. Now you can see there are different terms which have been mentioned here, particularly the inclusive fitness, direct fitness, indirect fitness. So one should understand these terms. One should have the clear conceptual understanding of this behavior in order to solve this problem. So let me just briefly discuss what these terms mean. Direct fitness is the personal reproductive success measured as the number of offspring that survive and reproduce. Means an individual, how many individuals it reproduces, how many of them survive and reproduce, that is something which is shown as its direct fitness. There is something called indirect fitness, wherein the reproductive success of individuals that share genes with the donor. That means, though they are not directly your offsprings, but the individuals we do share the genes with, they come under your indirect fitness. So all your niece, nephews, grandchildren, they do come in your indirect fitness because you do share genes with them, though they might not be directly your offspring as such. The term inclusive fitness comprises of both, that is direct as well as the indirect fitness. So here they have given all three things. They are asking you to calculate inclusive fitness, but they have provided you data for direct and indirect fitness in terms of number of offsprings which are directly produced by you, and in terms of number of nieces and nephews which have been saved by you, right? So the offsprings which are yours, they are coming under direct fitness. The offsprings of your siblings, that is niece and nephews which have been saved by you, they come under indirect fitness, and the overall fitness is called as inclusive fitness, which you are supposed to calculate. Now the question is, how do we calculate this inclusive fitness? How we can use mathematical formula to calculate this kind of fitness? There is a simple way. You need to take the number of offsprings which have survived. These are your offsprings which have survived and reproduced. Multiply it with the coefficient of relatedness, which is being shown by R here. So the coefficient of relatedness between you and your offspring, that is parent and offspring, you multiply the number of offspring with this R, and you add to it indirect fitness, that is number of non-descendant kids. That means which are not directly your offspring, but your kins. That means your family members. And multiply it again with the R, that is coefficient of relatedness. But this R value depends on the type of relationship that these kins are having with you. Now, going by this formula, we should know what do we mean by this coefficient of relatedness and how do we have a number or value for this R. R, or the coefficient of relatedness, is nothing but the proportion of alleles which are shared between two individuals as a result of common descent or common ancestry. So if there is a common ancestry, if there is a lineage, how much is the sharing of alleles between you and that particular individual? That is represented as coefficient of relatedness. If the relationship is between parent and offspring, R is 1 by 2 or 0.5 because half of the genes, half of the alleles of parent goes to the child. So child gets half of the alleles from the paternal side, half of them are from maternal side. So as one of the parent, half of your alleles have gone to the child. That is why the coefficient of relatedness is 0.5 or 1 by 2 here. If it is grandparent and grandchild, the R value goes down to 0.25 or 1 by 4. Similar is the case if the relationship is aunt and uncle and nephew and niece. Same ways, it goes to 0.25 or 1 by 4 in this case as well. So if you just remember these three values of R, you can solve this problem by using this formula. So in this case, the direct fitness calculation, that is the first part, number of offspring survived is being given as 5. You multiply it with the R between parent and offspring, which is 0.5. Add to it the indirect fitness, that is number of non-descendant gene, that is niece and nephews, that is 5 here. Multiply it with their R value, that is 0.25. So if you calculate this, you can easily arrive to the correct option, which is 3.75 in this case. Now, as I said, this is a very interesting problem, and similar kind of problems have been asked continuously in all the exams based on the altruistic behavior, based on the fitness concept, etc. So this is one type of problem that I have discussed. Similar to it is another problem which has come in December 2014. Now, this problem is also very similar, but a bit different on Hamilton's rule of altruistic fitness. Let's read out the question first. Assume that individual A wants to do an altruistic act to individual B, and that the benefit and cost of doing this act are in fitness units 40 and 12 respectively. According to Hamilton's rule, A should perform the altruistic act only if B is his, and then the 4 option is there. So now they have given the cost of performing an altruistic act, the benefit that recipient is going to have out of it, the value of that has been given, and they have given the 4 relationships. They are asking which relationship should he perform the altruistic act for. Remember, altruistic acts are done only when you have some sort of benefits out of it, though not necessarily direct benefit, but indirect benefits should be involved. And for that, there is a rule which is called as Hamilton's rule, which has been given like this. It shows the conditions under which altruism evolves. That means the conditions under which altruistic acts can be performed is given by this relationship. RB must be greater than C, where R represents the genetic relatedness. That means the kind of relationship for which you are performing the altruistic act. B is the fitness benefit to recipient. C is the fitness cost to altruistic. As you know, the altruism is something that you sacrifice yourself and you try to benefit the other members. So that means you have certain cost associated with it, which you bear so that it can benefit the recipient for which you are doing some altruistic act, right? And according to this condition, if you go, the value of RB should be more than the C. That means the benefits which you are getting for that particular relation should be more than the cost you incur. It's just like a normal business practice. The cost which you incur for a particular thing, you should have gains out of it. That means in order to achieve some benefits out of it, then only you are supposed to sell it. So in this question, value of B, that is benefit which recipient is going to have, has been given as 40 fitness units, the cost of doing this altruistic act is 12, right? And the relationship, you already know, the parent offspring, we have seen it's 0.5, grandparent grandchild, it's 0.25, aunt, uncle, nephew, niece, it's 0.25. So let's try to solve these four options based on this equation and see which is the correct option. For option A, sorry, 1 and 2, that is nephew and niece. Remember, there is no difference in terms of altruism, whether it is for nephew or for niece. They are simply your siblings' child. So both nephew and niece are having the same genetic relatedness with you. And the value of R for uncle, aunt, and nephew, niece is 0.25. Value of B and C we have already seen are 40 and 12 respectively. So if you calculate the RB, that is 0.25 multiplied by 40, comes out to be 10, which is not more than the value of cost, that is C here, which is given as 12. Hence, these two options are not the valid options. Same ways, if you go for option number 3, which is for grandson or granddaughter, if you calculate the value of RB, again, since the value of R is 0.25 again in this case, the value of RB comes out to be 10 again, which is more than the cost value, that is 12, and which is not more than cost value, which is 12, and hence this option should also be neglected. Coming to the option 4, whether it is for daughter or son. So the value of R here is 0.5, and you multiply it with the benefit, that is 40, you get the value of 20, which is more than the value of cost, that is 12, and hence this seems to be the correct option. So this person should carry out this altruistic act if it is for its own daughter or son. Then only it will give benefits or in terms of fitness. Otherwise, if it is nephew or niece or grandson or daughter, the cost is more than the benefit, and hence this should be rejected. So option 4 seems to be the correct one in this case. Okay, so moving ahead, this is question number 6, which is from December 2017 C part, and they have given a long stem. Let me just read out it quickly and then come to the conclusions. Orchids of the genus Cryptostylus are known to maintain reproductive isolation because their flowers look and smell like females of the wasp of genus Lysopimpla. When the male wasp visits and attempts to mate with the flower, the shape of anther and stigma allows correct placement and transfer of pollen to the wasp, which then transfers the pollen to species-specific flower that it next attempts to mate with. This prezygotic barrier that prevents interspecies cross-fertilization in Cryptostylus is best explained by, and then the 4 option is there. Now, you can easily make out this question is from the isolating mechanisms and the barriers part. So one must have the basic understanding about these kind of barriers or isolating mechanism. You might have read various isolating mechanisms. Here, this one, I'm just giving you a snapshot of Meckham's classification, which has divided the barriers in pre-mating or pre-zygotic and post-mating and post-zygotic type. And at each step, the barriers are there. The first step, potential mates do not meet, and the type of barriers which come under there. Potential mates meet but do not mate with each other. That kind of barriers can be there. They can be the barriers where the copulation is attempted, but the transfer of sperms does not take place. So at each step, if you just consider pre-zygotic here, the barriers are existing. Now, in this question, 4 barriers have been mentioned: the seasonal or temporal one, habitat barrier, ethological or behavioral barrier, and the mechanical isolation. So we must understand what do we mean by these barriers. Now, this is a busy slide. I don't want you to go in detail of it. Let me just quickly make you recall what all these barriers are. Habitat isolation is when the two species occupy different habitats. That means there are occurring in two different habitats so that they do not actually come close to breed with each other. Temporal means they breed at different times of day or seasons or years, and hence the barriers is existing. Ethological or behavior is the courtship behavior particularly. They are unique to a particular species. So two different species do not recognize the courtship behavior for mating. Mechanical isolation means we have different and complex structure of genital organs in different organisms, which does not allow the copulation to take place between members of two different species. Now, if you have this basic idea and you focus on this question, ignore the left part. I have just kept it to remind you of the question, but focus on the right side. Two lines are important in this question stem. First, they are asking because their flowers look and smell like females of the vase. So the concept of mimicry is already there, right? So one should focus upon that. Second thing they are saying, the shape of anther and stigma allows correct placement and transfer of pollens to the vase, which then transfers the pollen to species-specific flower that it next attempts to mate with. Hence, a component of mechanical compatibility is mentioned over here. Then, if you just read out these options, let's go by fourth option, habitat isolation. Nowhere in the stem question stem they have mentioned that there is breeding at different time of the year or season or day, right? There is no different habitats as well, right? There is no behavioral isolation as well. There is no courtship behavior involved. There is no different type of behavior associated with the mating. The second option, which is mechanical isolation through mimicry, seems to be the most perfect one in order to be consistent with this question scenario that has been presented. So the right option here seems to be the mechanical isolation because there is the mechanical compatibility of transferring the sperms or anthers to the other organism through the wasp, which is showing the component of mimicry. So mechanical isolation through mimicry seems to be the correct option here. Okay, moving ahead to question number 7 here, which came recently in June 2019. They have depicted a phylogenetic tree of selected taxa, and they're asking to choose the correct statement out of the 4 options. Now, this is again from the phylogenetic trees. How we interpret the phylogenetic tree, based on that, you need to choose the correct option. There are different terms, as you can see, the monophyletic, polyphyletic, paraphyletic, etc. One should be clear about how we interpret the phylogenetic trees and what these terms depict or represent in order to solve this question. So the first term, which is there in monophyletic, it means a group which is made up of an ancestral species and all its descendant species. So here, if you see, B is the ancestral species, and all these, that is C, D, E, G, H, F, they are all its descendants. So ancestor along with all its descendants, when they are clubbed together in a group, that is called as monophyletic, right? In contrast, you have paraphyletic, wherein you do have a common ancestor, but not all the descendants are included in the group. Instead, some of the descendants are included. For example, here, A is the common ancestor. Its descendants are on the both sides, that B and its descendant are also in descendants of A. I, J, and K are also descendants of A, but in this group, they have only shown I, J, and K as the descendants of the ancestor A. Means the common ancestor has been marked along with the sum of its descendants. Such kind of grouping is called as paraphyletic. In contrast to monophyletic, where all the descendants were marked, here only a few of descendants of the common ancestor have been grouped. Third category is called as polyphyletic. It is polyphyletic group where there is a lack of the common ancestor. Means FGH, IJK have been grouped together. There are two different clades and different taxon are there, but common ancestry has not been involved. Neither B nor A has been involved. So if you just have the group of taxon or different taxa without involvement of common ancestry, then that particular group is called as polyphyletic. Coming back to this question, again, I would request you to focus on the right side. If you see group X, there, this ancestral species has been shown. Here is the ancestral species, that is the branching point or the origin point. And all its descendants have been marked, that is A, B, and C, which is coming from this particular origin. That means this is a monophyletic, as we have seen in the previous depictions. If you focus on group Y, B, E, F, and G, B, E, and F have been grouped together with a common ancestor, and G has been excluded out of it. So this is the case of paraphyletic, where we include the common ancestor and some of its descendants, and we do not include all the descendants of that common ancestor, and hence this is the paraphyletic. So if we are clear about this, we can easily see correct option here seems to be option number 4. Group X is monophyletic and group Y is paraphyletic. So we can easily reach to the correct option based on this kind of understanding. Now this question number 8 is sort of cladogram. It is a type of phylogenetic tree only, but then this is more of analysis of the given options. So it's more of interpretation of the phylogenetic tree, how you interpret it. Based on that, you can easily reach out to the correct option. It says, with reference to the phylogenetic tree presented above, which of the following statements is true? Some of the statements have been made seeing at the tree, and they're asking you to select the correct option. Let's take each option one by one. Option 4, if you see the tree and just focus on option 4, it says lampreys and mammals are not related. Then try to see the lampreys and mammals in this question. The option says they are not related, but this is not true. We can say that lampreys and mammals are distantly related. They are distant apart. They are farly distant, but we cannot say that they do not relate with each other, because you can see here there is a common ancestry. So all have evolved from this common ancestor, so they are in one way or other related with each other. There might be difference how closely or distantly they are related. So this option does not seem true. Third option is cartilaginous fishes are the ancestors of amphibians. If you see cartilaginous fishes and amphibians in the phylogenetic tree, we cannot say that cartilaginous fishes are the ancestors of the amphibians. They do arise from a common ancestor. They have evolved independently, but amphibians are not the descendants of cartilaginous fishes here, right? So again, this option is also rejected. Second option is birds are more closely related to reptiles than to mammals. Has it been the case, birds and the reptiles should have been put together with a common ancestry rather than birds and mammals as shown in this phylogenetic tree. So this option again seems to be incorrect one, because birds and mammals, they do arise from a common ancestor and they are more closely related as compared to birds and reptiles. And hence, this is again the rejected one. So ultimately we reach to the first option, amphibians, reptiles, birds and mammals do share a common ancestor. As it's very much clear from this phylogenetic tree, all these groups do have a common ancestry and they do evolve from a common ancestor, and hence we can say that this is the correct option. Okay.
Hello, Sudhir sir.
Yeah.
Uh,
Yeah, I can see the time. It's already 10:55. So should I stop here?
we'll wait for a minute. If there are some questions, we can take up the questions.
Definitely. So let me just stop sharing the screen and take
Yes,
the
sir.
question first, and then we'll come back to this.
Okay.
Yeah. So please let me know if there are any questions.
Yes, sir. Yes. I guess the way you have explained very meticulously, participants seems no questions as such.
Okay. All right.
Still, Javan sir, is there any question in the YouTube? I have gone through it, but something if I missed, you can tell me. Jain sir. I guess there is no response from Jain sir also because
Okay.
in YouTube I don't see any question as such, because the way you have elaborated each question itself is a clear understanding to our participants. Okay.
Okay.
Sir,
So still there is a question.
there are a couple of questions.
Okay. Fine.
Okay. I can read one question.
They are asking about reference books.
Your voice are echoing. Can everybody else, please mute your mic so that I can hear the question properly?
So they are asking about reference books.
Okay. If you're asking about the reference book for evolution, particularly, I would suggest the book by Strick Burgess, Evolution, that is by Hall and Hall Grimson. It is one of the very good one. Besides that, you can also refer the book by Futuyama, and also there is a book by Ridley, which is also very broadly used in evolutionary concepts. These are three prominent books which I can refer to you for evolutionary biology. I would also suggest you to go through the video lectures. These are the video lectures which I have developed through Swayam Prabha only for evolutionary biology, which is a sort of crash course kind of thing to revise all the concepts of evolutionary biology. It's a 17 hours course only. So I'll share the link of that course, which you can share on see on the YouTube, and you can go through those lectures also for having a clear understanding of the basic evolutionary concepts.
Okay, there is a question.
Yes.
Why is genetic drift more prominent in small population?
All right. So why genetic drift is more prominent in small population? Okay. See, if you read the concept of genetic drift, basically there is a change in it's a microevolutionary principle or change, which brings out change in the allele and genotypic frequencies. So if a population is small, right? Let's say if there are only 10 individuals in the population, for an example, if we take, and let's say there is a bottleneck effect. That means there is a natural calamity because of which only a few numbers are left, or there is a founders effect, any of these. And let's say out of 10, 7 individuals die off. So only 2 or 3 individuals are left. So there is a drastic change in allelic frequencies because of it. In contrast, if you have a large population, that means, let's say, if there are 1,000 individuals, right? And then there is a natural calamity through bottleneck effect or through founders effect, if there is any migration of individuals, still there will be sufficient number of individuals which do not cause a large amount of change in allelic frequencies. So this is the reason why we have small number of uh, population number shows prominent effect in terms of change in allelic frequencies or the genetic drift compared to the large numbered population or the large population.
Okay. Thank you, sir. Another question, what is a realistic example of genetic drift?
See, as I just mentioned, genetic drift, two of the realistic examples can be either through natural calamity or through a migratory events. So if there is a natural calamity, as I just mentioned, if most of the individuals die off, so there is a sort of bottleneck. You can correlate bottleneck with just a traffic jam, right? When we are moving to the road and there is a jam and you come to the point that there is a narrow point ahead because of which one by one vehicles are moving because of this bottleneck, there is a slow speed. So similar situation we can have if there is a natural calamity, right? And out of a large population, if it is there, most of the individuals die off, which results in a small population and change in the allelic frequency. So that is one natural example. Second thing, if there is a large amount of migration, so there is a founder effect. That means some of the individuals of population move out and go to some other place, and there they establish a different population or different colony. Then also there will be a drastic change in the allelic frequencies. So these two events do occur realistically or in the natural population, which bring out the genetic drift.
One more question, how to remember geological timescale using trick?
Okay, see, just like our school days, we used to make acronyms, right? So for geological timescale, what I use is the acronym, right? So going from eons to era to periods to epochs, for each one, you can make an acronym like if you have Mesozoic era, Cenozoic era, etc. So you just take first alphabet of like M, C, etc. and you make an acronym word and try to remember it. That is the only way we can remember all these large cumbersome names which are there in geological timescale, and it applies to remember the names in case of, let's say, horse evolution or human evolution. That is what works, and I usually use that. So make the acronym and try to remember that.
Okay, thank you very much, sir. These are the questions which we have received directly. At the outset, I like to thank you for your overwhelming and very clear distinction about the question asked in the recent years in the CSIR rate. And I expect and I have guarantee that all the participants, they have got the clear idea about the concept. Thank you very much, sir.
My best wishes to all the participants. Thanks a lot.
Thank you, sir. Now we have to break for 2 minutes. Then we'll join after 2 minutes. Hello.
Sir, am I audible?
Yes, you're audible.
Thank you.
Yes, sir. Yes, sir. After 2 minutes, we'll start.
In case you can share that slide of tea break.
Okay, good morning, everyone. We'll be starting with the second lecture by Professor Kaushik Inamdar. I'm happy to introduce him at the outset. Professor Inamdar, just a minute, I'll share the slide. Professor Kaushik Inamdar is MSc Microbiology from Mumbai University. He is a SET qualified and at present is pursuing his PhD in microbiology from Mumbai University. He is at present assistant professor and head of life science department in KSG's Model College, Dombivli. Earlier, he has worked with KV Pendarkar College, Dombivli, and he has also an industrial experience. He has worked in Cytex Lab Private Limited, which is a firm of CIPLA. He has a good number of publication on bioremediation. He also has guided UG projects, research projects, and very important is being a science person. He is a very good Marathi poet. He composes poem and he recites poems, and he's been invited in many places for his elocution, his speeches and poems. I hearty welcome, sir, and
Thank you so much.
session is yours. I'll remove the slide.
Very good morning, all of you. Okay, I'll share my screen first and then I'll talk. Is it visible, all of you?
Yes, sir. Yes, sir. It is.
Yeah, thank you so much. So good morning, everyone. Today, I'm taking applied biology. Before going to the topic, let me share something with you. First of all, I want to say thanks to INEAS and ATBS for giving me this opportunity to interact with NET set aspirants. Okay, now let's come to applied biology. What is applied biology? So students, you can post your answer in the chat box. Simultaneously, I'm going to the YouTube also. Okay, so I'll get your answers here. So post your answer. What is applied biology? Okay. Yeah, I can see some answers. Okay, great. So applied biology basically it focuses, okay, on the practical use of biological material. It focuses on the practical use of biological material. Now, which type of biological material we can use? See, biological information, biological material can be anything. It can be whole organism, it can be bacteria, it can be virus, it can be DNA, it can be enzyme, it can be protein, it can be anything. Okay, so application is must. Now it can be, we can say the production of secondary metabolites. So I hope you have studied secondary metabolites produced by bacteria, actinomycete, fungi, yeast. Okay, so this is also important. Then production of antibiotics. So all of you know penicillin, the best antibiotic produced by Penicillium fungi. Okay, so production of antibiotics, production of vaccines, transgenic animals, plants, then use of microorganisms to eat the waste. See, this is the need of today, of the future, to produce such microorganisms or to search for those microorganisms which can eat plastic, which can eat oil spill degrading bacteria. Okay, that is the need. So this comes under applied biology. Now, when you talk about students net and set, okay, this is a very important exam. Now, weightage point of view, if you see applied biology, okay, it is the fourth most important topic. So the weightage, I think, is approximately 6 to 8%. So you will find questions in B part as well as in C part. Okay, so weightage is approximately 6 to 8. So high weightage is there for applied biology. So it is very important topic regarding NET and SET. Now, one more important thing which I would like to share, I would like to stress that study for paper 1 as well. Now, generally being a bio student, okay, जब हम 12th में होते हैं या जब हम पढ़ रहे होते हैं biology, okay, it can be microbiology, zoology, botany, life sciences. Generally, we have fear about maths. Okay, generally हम दूर भागते हैं maths से. Okay, but it is important for clearing net set, and it will surely help you in clearing net set for getting higher marks. Okay, and it is not that much difficult, students. Okay, it consists of some topics like logics, probability, profit loss, numbering, which you have studied in your school level. Okay, so study paper 1 as well. Okay, now let's go to applied biology. Now this is the NetSet syllabus, CSIR NetSet syllabus for applied biology. So following topics, as you can see on the screen, topics are included in the NetSet syllabus. So today we will study these headings one by one with one or two questions. Okay. So I am going to solve the questions for you, and I want you also, okay, to take your pen and paper. Okay, jahan bhi ho, jao, pen and paper leke aao and try to solve it. Okay. So I will be solving, don't worry. But along with me, you have to solve it. You have to think on it and post your answer in the chat box. Okay, that is important. Now, the syllabus consists of microbial fermentation, products of microbial origin, vaccines, transgenic animals, plants, genomics, markers, bioremediation, and biosensors. So, number of applications you will find in this applied biology syllabus. Now, reference books are also given, as you can see in the slide. Reference books are also given, and you can refer this. You will get a lot of information for all the headings which are there on the screen, okay, from this reference books. I'm very much sure about it. Okay, so let's go to the first topic. And yes, it is very important. I forgot to tell you in the first slide, it is very important to solve last year's question papers. Okay, if you solve last 5 years question papers, I am very much sure you will get confidence. Okay, I will not say that you will clear the NetSet 100%. You will get the confidence that yes, I can clear NetSet examination. Okay, so solve last year's question papers. Now let's go to the first topic, that is fermentation. Microbial fermentation and production of some molecules. Okay, now in this, fermenter design is very important. Fermenter design and types. So in the types, there it can be batch fermenter, it can be continuous fermenter, it can be say packed bed, it can be airlift, stir tank. So there are number of examples, number of types of fermenters. Generally, इस पे ज़्यादा questions नहीं आते हैं, but still this is important. Next point, products of microbial fermentation. This is very, very, very important. You will surely get one or two questions in net paper from this. That is production of beer, organic acids. So organic acids can be lactic acid, citric acid, okay, vitamins, amino acids, okay, antibiotics. It can be penicillin, streptomycin, vancomycin, and so on. Then number of, we can say, therapeutic proteins are there. Okay, so this might become in the examination. Okay, now let's go to the first question. So the question says the following table. Okay, I'll just try to solve this. Okay, so the question says the following table lists some of the enzymes. Okay, some of the enzymes of fungi and bacteria having wide variety of industrial applications, including alcoholic beverages, food, detergents, and pharmaceuticals along with their microbial origin. Okay, now let's solve this question. Now let's first go to the, so the column A consists of enzymes and column B consists of microorganisms. Okay, now let's go to the D part first. Okay, so the first option, the correct combinations are, so there are 4 options and we have to select one out of. Okay, let's check the one. Okay, D and F. Now D and F, Serratia marcescens and pectinase. Am I audible properly? Hello, am I audible properly?
Yes, sir. Now
yeah. Okay, thank you. So when we talk about Serratia, a beautiful microorganism, I must say, it produces beautiful red color colonies on agar. So Serratia is generally a human pathogen, students. Okay, and when we talk about pectinase, so pectin is generally a outer covering. It is present in the outer covering of fruits. Okay, so it is not producing pectinase type of enzyme. So this option is wrong. Now let's go to the next, B and G, that is asparginase and aspergillus. Yes, अब यहाँ पे नाम का similarity है, asper. Okay, so you may get confused in this, but asparginase, and for these students you need some knowledge of microbes, and their products, their main products at least. Okay, so asparginase is enzyme basically produced by E. coli and Aspergillus niger. Okay, so Aspergillus niger, a fungi, a very beautiful fungi, it produces asparginase as a main product, but not Aspergillus aureus. Okay, so this is also ruled out. This is also, let's rule out this. This is also ruled out. Now let's go to the third, that is C and E, lipase and azotobacter. Now azotobacter, ये नाम आपने बहुत बार सुना होगा. It is a nitrogen fixing bacteria. Okay, nitrogen fixing bacteria. So the major enzyme कौन सा होना चाहिए? It has to be nitrogenase. Okay, so majorly it produces nitrogenase enzyme and not the lipase. Okay, so this we can rule out. Okay, now let's come to the fourth option. Okay, A and H. Let's confirm this. Okay, let's confirm this. Now, when we talk about Aspergillus oryzae, Aspergillus oryzae is very important fungi. उसको हम कहते हैं koji. Okay, so this koji produces alpha amylase. This produces alpha amylase as a major product. Okay, so Aspergillus and amylase is the correct option. So the answer is 4. Okay, so in this way you have to deal with the questions which are regarding the industrial fermentations. See, here you need some knowledge, you need some understanding about microbes, about their products. Okay, so that is very, very important. Now let's go to the next. That is, the next topic is the application of immunological principles and tissue culture method. Now this is also very important. Okay, tissue culture. Now let's come to the vaccine first. So vaccine is also an important topic, especially the types of vaccines. Okay, so heat killed and attenuated. Okay, DNA vaccines. Okay, so these types you should study, because one or two questions I have seen on the vaccines types. So it is also important. Now let's come to the plant tissue culture. So in plant tissue culture, PTC media, plant tissue culture media is important. In that also hormones is important topic. Okay, so it can be 2,4-D, auxins, cytokinins, gibberellins, ABA. Okay, but they are important. You will find these questions in last 4 or 5 years question paper. So this is important topic. Then culture types. So it can be, we can say protoplast culture, cell suspension culture. Okay, so these topics are also important. And last, but not the least important, is somatic embryogenesis, plant regeneration. Okay, callus का कैसे plant बनता है, plant regeneration, somatic embryogenesis, especially the stages. Okay, so stages are very, very important. That is 2 cell to 8 cells, 8 cell to say 16. Then we have a heart-shaped embryo. Then we have a torpedo-shaped embryo. So that is important in plant tissue culture. And when we talk about animal cell culture, again the media and the growth requirements are important. Okay. Now let's go to the question which is based on the plant tissue culture only. Okay, so the following statements are related to plant tissue culture. So let's read out the statement first. Friable callus provide the inoculum to form cell suspension. The process known as habituation refer to the property of callus losing the requirement of auxin and cytokinin during the long-term cultures. Cellulase and pectinase enzymes are usually used for development. Torpedo stage is formed before the heart stage embryo. Okay, that's why I told you this embryo development is also important. Now, which of the following combinations of the above statement is correct? Let's find out. Okay, so let's first go to the first, that is friable cultures. Now, when we talk about cell suspension cultures, now it generally fall in 2 categories. Okay, either compact or friable. See, as you can see in the picture, this is a compact one and this is a friable one. Okay, so compact one friable one. Okay, so in the compact one, cells are tightly aggregated. Okay, in the friable, they are loosely aggregated in the callus. Okay, so generally, as they are loosely aggregated, as you can see in the picture, they can generally break apart. Okay, and when they break apart, we can use them for cell suspension cultures. Okay, so the first option, that is friable callus, provide the inoculum to form cell suspension culture, is true. Now let's come to the second, the process known as habituation. Now, when students, when you learn on when you study plant tissue cultures, habituation is, we can say, a process where a callus lose something. Okay, and especially it lose some, we can say, hormones which are required for plant regeneration and plant development. Basically, important hormone is cytokinin. So generally, they lose the requirement for cytokinin. Okay, so this statement is also correct, true. Now, third is important. You should know something about protoplast cultures. Now, what is protoplast? Yes, protoplast is nothing but the plant cell without cell wall. Okay, so when the plant cells lose its cell wall, it is called a protoplast culture. So protoplast culture is also very important in genetic engineering. Okay, now how we can remove the cell wall of the plant cell? So we can use mechanical methods, we can use chemical methods, we can use enzymes for it. Okay, so generally plant cell, if you see, it is made up of cellulose, pectin, and number of other ways. So if we use cellulase, which degrades cellulose, and pectinase type of enzymes, it removes the cell wall, okay, and thus create a protoplast. Okay, so this is also true. Okay, now let's come to the fourth option. Now, during somatic embryo development, torpedo stage is formed before the heart stage. Now let's see this picture. Somatic embryo development. So as you can see in the picture, the cell, a single cell, divides to form 2 cell stage, to form 8 cell stage, to form 16 cell stage. Okay, and then after you will form a globular stage in the somatic embryo development. After the globular stage, there is a heart stage. Okay, and heart stage, in this you will find cotyledon development. Okay, and because of the cotyledon, see, globular stage mein koi cotyledon development hota nahi. Cotyledon development will start in the heart stage after the globular stage. So because of the cotyledon shape, generally it we can say wo heart shape dikhta hai. Okay, so heart stage after the globular, and after the heart stage, this cotyledon will develop. Okay, and it will form a torpedo like structure, torpedo stage. So heart stage comes before the torpedo, and torpedo comes after the heart stage, of course. Okay, so let's go to the fourth option. During somatic embryo development, torpedo stage is formed before the heart stage. So torpedo stage comes after the heart stage. Okay, so this option is wrong. Okay, so which one is correct? A, B, and C. So let's see. Option A says A, B, and C. Option B, A, B, D. Option C, B, C, D. And option D, A, C, D. So the correct one is A. That is A, B, and C. Okay, so in this way you have to solve the questions regarding plant tissue culture. Okay, so this is important. Now let's go to the next. Vaccinations, yes, important. Now given below are the type of vaccinations in the column A, diseases in the column B, and we can say the advantage or disadvantage of it in the column C. Which of the following combination is most appropriate one? Okay, it is asked again in the 2018 net. Okay, so let's see. A column says live attenuated, inactivated or killed, and inactivated exotoxin. Option B, rabies, measles, and diphtheria. Option C, strong immune response, often lifetime, stable immune response, and chance of untoward immune reaction, less successful, and repeated administration. Now, see, before attempting this, you should have some knowledge regarding vaccines. Okay, it's a very important topic in immunology as well. Here also it is important. Okay, but we can solve this with some tricks. Okay, some faster way. Okay, you should have some understanding, you should have some knowledge of microorganisms. Now, being a microbiologist, first I'll go to exotoxin. Okay, let's go to C, exotoxin. So inactivated exotoxin. So here exotoxin word is very important. There is only a single microbe in the column B, students, which produces a very potent type of exotoxin, that is a diphtheria toxin. Okay, so when we talk about exotoxin, diphtheria toxin play a very important role, and we all know that. So let's search for an option which contains C and 3. So option A contains C3X, option B contains C3Z, option C C1Z, option D C1. Okay, so first and second contains C3, so we can rule out C and D option. Now let's concentrate on A and B. Okay, now let's see this classification of vaccines. Now when we see the classification, it can be whole agent, it can be subunit. Now subunit को थोड़ा बाजू में रखते हैं. Whole agent vaccine, when we talk about whole agent vaccine, majorly it can be inactivated vaccine, killed vaccine. Okay, kill हम किससे करते हैं? Either with the help of formalin or heat treatment. Okay, or attenuated. Attenuated is removing the pathogenicity, not the immunogenicity, just the pathogenicity. Okay, so the example for inactivated kill is rabies, and attenuated का example है MMR vaccine, measles, mumps, rubella vaccine. Very important vaccine. We should generally administer करते हैं हम लोग starting from child age to age of 15 and 16. 2 or 3 doses are required for MMR vaccine. Okay, so let's go back to the. Okay, now let's go to measles. Let's go to measles. Now measles vaccines, generally if we administer this vaccine, it gives you long-term immunity, lifelong immunity. Okay, generally आपको measles होता है, okay, या rubella होता है, life में आगे कभी भी होता नहीं. Okay, so measles vaccine also give you a very long-term immunity. What is the reason for it? कौन सा disease causing agent है? Measles का paramyxovirus. This paramyxovirus is we can say it do not mutate rapidly. So जैसे हम अभी कोरोना वायरस में देख रहे हैं, okay, mutants आ रहे हैं, बहुत सारे mutants आ रहे हैं, and thus हमारे ऊपर एक question है कि vaccine work होगा नहीं होगा. But in measles, the vaccine will work because the mutation rate is not that much higher, so it will give you lifelong immunity. So let's search for the option. So A option say that A1Z, A1Z. Okay. B option says B2Y, B2 and Y. Okay. So हमने अभी क्या देखा? Measles gives you lifelong immunity. So 2 option should go with X. Okay. So let's search for 2X. So B option has A2X, A2X and B1Y. Okay. So rabies is always, remember, it is killed. Okay, never it is attenuated. So the option B is correct. Option B is correct. Okay. So in this way, you have to solve vaccination type of questions. Now let's go to the third part, that is transgenic plants and animals. Okay. I'm going through the chat box also. Many of you are attempting it correctly. Very good, students. Transgenic plants and animals. So in this, with respect to transgenic animals, construction of transgene and delivery is very, very important. So you can use micro-injection, viruses for inserting it. Transgenic animals, basically. In transgenic plants, gene transfer methods are very important. So mainly 1 or 2 or 3 or 4 questions are also expected in the paper. Now, in direct methods, you will see gene gun, micro injection like that. But very important. Okay, production of transformed plants with Agrobacterium tumefaciens and TI plasmid. This part is very important. Okay, so if you have to study in this, you have to study basically we can say tDNA, TI plasmid, okay, virulence genes, VIR genes. That is very important. Okay, so let's see one question regarding this production of transformed plants with Agrobacterium. Students, I'm again telling you, this question, this topic has a very good weightage. You will find one question at least from this in every paper. Okay, you go to any paper right from 2010 to 2019. Okay, you will find one question on Agrobacterium tumefaciens. So it's very important. Let's solve the question. Agrobacterium tumefaciens is frequently used as a vector, okay, to create transgenic plants. Yes, true. Under laboratory conditions, Agrobacterium mediated plant transformation does not require. Okay, मैंने यहाँ पे bold क्यों किया है? Because generally we miss out this. Does not require. Okay, so options are host plant genes, bacterial type 4 secretion system, virulence genes, VIR genes basically, and opine catabolism. Okay, now let's go to the next slide because we need some photographs, we need some information to solve this question. Okay, so Agrobacterium tumefaciens is generally used as a vector, right? So it contains TI plasmid. As you can see in the picture, TI plasmid. So plasmid contains tDNA, where you can insert a foreign gene. So generally it is used for genetic engineering, okay, transformation of plants. So we have to insert some gene, foreign gene into it. So foreign gene is inserted in the tDNA, okay. It contains virulence genes responsible for virulence. So virulence matlab because of these virulence genes, they can infect the plant cells. Okay, that means it is also important. Let's go back to the so does not require. So plant transformation, what question says? Plant transformation does not require. So virulence genes are required. Just now we have studied. So virulence genes are required. So this is not the answer. Okay. Now, then after infection, basically, tDNA from the plasmid transformation, okay, with host genome. So host plant genes are required. See, it is going to infect the host. It will get integrated into the host genome. Okay, so host plant genes are very required. So A is not the answer. See, A is not the answer. Now let's go to the B, bacterial type 4 secretion system. Now what is it? Now when we talk about tDNA, okay, as we are saying, it will enter into the plant cell, it will integrate into the host genome, but how? Okay, there has to be some medium, there has to be some bridge. Okay, that bridge is nothing but type 4 secretion system, okay, to transfer the tDNA into the host cell. Okay, so type 4 secretion system is also important. Okay, type 4 secretion system is also important. So this is not the answer. Okay, so opine catabolism genes. Now हमने तीनों को ruled out कर दिया है. So probably answer can be opine catabolism genes, but let's confirm that. Okay, confirmation is very important. Just ऐसे ruled out करके नहीं चलेगा. So let's confirm that. See, you always use, see, my funda of your solving the question to eliminate, okay, the non-probable answer. जो हो नहीं सकते उसको eliminate कर दो and then think about the probable options. Okay, so you can go with the same or you can choose your own method, but this elimination or strategy works many a times. Okay, so let's discuss opine catabolism. Opine, what is opine? Opine serve as a carbon and nitrogen source for the bacteria. Okay, so it is required. Okay, but in the question, if you see question, what is written? What is written? To create transgenic plants under laboratory conditions. This is very important. So we are not going for actual infection of plant. It is in the laboratory condition we are doing. So in the lab conditions, okay, as we are providing all the nutrients, so opine catabolism genes are not required, because opine is what? It is a carbon nitrogen source for the agrobacterium. We are already providing it in the media, so opine catabolism genes are not required for agrobacterium to grow. Okay, so that's why the answer is D, opine catabolism genes. Okay, let's go to the next. Next topic is breeding in plants and animals, including marker assisted selection. Now, what is marker? Okay, basically when we talk about molecular markers, it is nothing but a fragment of DNA. Simple karke bata raha hu, it is a fragment of DNA that can be identified in the genome. Okay, so it is a specific fragment of a DNA which is there in the genome, and we have to find it out. It is a molecule, it serves as a molecular markers. Okay, so molecular markers we can find out with the help of RFLP, RAPD, SSR, okay, AFLP, SNP, etc. There are a number of ways, like RAPD is random amplified polymorphic DNA. Then we have RAPD is random amplified polymorphic DNA. RFLP, restriction fragment length polymorphism. SSR, simple sequence repeats. So these are all important molecular markers. Okay, and this also you will find 1 or 2 questions in every paper. So this is also important. Marker assisted selection and breeding also important. You will hardly find question on this, but this is also important. Now let's go to the question. Okay, so molecular polymorphic markers are already known with respect to tobacco mosaic virus resistance in tobacco. Among this, which marker system you will select that will be simple, economical, and less time consuming? Okay, so important is this last part. Okay, that is the simple, economical, and less time consuming. So all these 4 we can use. Okay, but which one is the simple, economical, and less time consuming? Okay, now let's go to all the 3. Okay, let's go to first EST SSR. Okay, now EST is nothing but if we want full form, EST is expressed sequence tags. Okay, so EST, these are the DNA sequences that can be used to detect basically coding sequences, okay, in the genome. And SSR is nothing but tandem repeats. Okay, now these play a very important role, EST SSR, in finding out the tandem repeats which are very specific for the coding regions. Okay, this method is the most reproducible method. This method is simple as well as economical. Okay, so let's isko mark karke rakhte. Let's go to RAPD. Now what is RAPD? RAPD and RFLP, we can say it is, when we talk about RAPD, due to mismatch annealing, the reproducibility decreases in RAPD. Okay, so mismatch annealing is very much common in RAPD, so the we can say reproducibility decreases. So we can rule out this option. Okay, as it is simple, economical, and less time consuming. Also the efficiency has to be maintained. Then when we talk about RFLP and AFLP, that are very complex and laborious techniques. Okay, so that can increase the cost also. So that also we can avoid. Now when we talk about EST SSR, again is better over rest of the 3. Okay, it is reproducible, completely automated. See, when we talk about RAPD and other things, RFLP, they are not completely automated. EST SSR is completely automated, so it is less time consuming, economical, simple as well because it is properly automated, and very efficient and reproducible as well. That's why the answer is D. Okay, you have to read all the RAPD, RFLP, AFLP, EST SSR to solve such type of questions. Now let's go to the next and last part, that is bioremediation, phytoremediation, and biosensors. Now this also very important. See, when we are living in the world where we can say dirt and garbage is increasing day by day, and there is need to decrease the garbage, decrease the contaminants, decrease the pollutants. Okay, bioremediation play a very important role in all these things. Okay, so bioremediation is nothing but, when we talk about bioremediation, it is an important tool, okay, to degrade the contaminants. Contaminant can be oil, contaminant can be dyes. Okay, so in this, generally microorganisms are used, and microorganisms use number of strategies to degrade these you can say contaminants and pollutants. So this play a very important tool. Now, microorganisms, you should know some names of microorganisms, strategies they use, like it can be biosparging, okay, bioaugmentation, okay, bioventing. So these are some strategies which we can use in bioremediation. Phytoremediation, as the name suggests, phyto, use of fungi. So fungi play a very important role, as they are saprophytes. Okay, they play a very important role in bioremediation. And last part is biosensors. Now, what is biosensor? Yeah, so it can be so see, biosensor, see, it can be anything. It can be an enzyme, it can be an antibiotic, it can be nucleic acid as well. Okay, so these things, they react with some analyte, and they will give you some response. Okay, so response, a signal that can be measured, and we will get an output. Okay, so that we can see the change, okay, over a specific period. Let's take an example. Okay, let's take an example. I'm a microbiologist, so I'm going again with the microorganisms. So let's take example of methane biosensor. Okay, so methane biosensor, again, why I'm taking this example, because it is coupled with bacteria. It is coupled with bacteria, okay, which will consume methane. Okay, and carbon oxygen also. So it is coupled with oxygen electrode as well. So when the bacteria will grow, okay, we can say the methane amount, as the methane amount increases, okay, the oxygen level will decrease, and as the oxygen level will decrease, you will get we can say less reading, or the current will start decreasing. Okay, so and that will, in the output, you can see a decrease in the reading. Okay, so you can plot a graph and you can see the result. So this type of biosensors play a very important role in number of we can say studies, research. Okay, let's go to the question. Let's solve this question together. Very important. In bioremediation by microorganisms, detailed below, choose the incorrect option. Again, incorrect. Okay, first is organic contaminant provide source of carbon. Bacteria do not get energy by degrading the contaminants. Bacteria can produce, oxidize, or reduce species. Bacteria can act on contaminant by aerobic and anaerobic respiration. Okay, now you must have studied microbiology, and you should know how microbial growth takes place. Okay, so let's go to this. Okay, so when we talk about microbes or microbial growth, they require some nutrients like carbon, nitrogen. Okay, so in this we are providing generally, when we grow in lab, we provide them in the media. Okay, so that they will grow. But when we are allowing them for bioremediation, okay, on some toxic waste, they use that carbon, they use that, we can say, toxic waste, or we can say any material which has to be degraded as a carbon source. Okay, so they need, every bacteria need carbon and energy source. Okay, carbon source and energy source is must. So they utilize carbon source, they utilize that toxic waste as a carbon source as well as energy source. Okay, so carbon, organic contaminants provide source of carbon. This is true. Bacteria do not get energy by degrading contaminants. Okay, isko thoda baaju mein rakhte hain. Bacteria can produce oxidized or reduced species. Now, bacteria can oxidize and reduce number of species. Okay, so yes, because they can they degrade pollutants now. So by how they degrade the pollutants? By either oxidizing them, okay, or by reducing them. So they oxidize some compounds, they reduce some compounds, and that's why they are degrading them. So bacteria can produce oxidized or reduced species. Now let's come to the fourth, that bacteria can act on contaminant by aerobic energy. Yeah, very good. Bacteria can act on contaminant either in the presence of oxygen or in the absence of oxygen. Some of the, let's take an example, okay. Okay, some anaerobic bacteria, especially methane degrading. So हमने अभी question देखा था methane degrading bacteria. So in methane degrading bacteria, example methanosarcina. Okay, so they efficiently grow basically in absence of oxygen, that is in the anaerobic condition. Okay, so they can also act on the contaminant in anaerobic respiration. Aerobic bacillus, okay, aerobic respiration say they can degrade the contaminant. So again, this is also correct. Now let's come to the second option. Bacteria do not get energy by degrading the contaminant. हमने अभी देखा था, okay, that bacteria will use the organic contaminant, okay, as a electron source, that is the energy source, as well as the carbon source. So they need energy and they get that energy from contaminant only, okay, because we are not providing any media requirements. हम कुछ इसमें कार्बन नहीं डाल रहे, हम इसमें कुछ पेप्टोन ऐसे कुछ नहीं डाल रहे. We are not giving any media requirement there. So they have to get the requirement, they have to get the nutrition from the contaminant only. So this option is wrong. Okay, bacteria get energy by degrading the contaminant. Okay, so in this way you have to solve the problems regarding bioremediation. Again, you will find one or two questions from bioremediation in the question paper. Okay, again an important topic. As a point of environment also, it is very, very important, students. Bioremediation, you should study, and more research is required in bioremediation area, because it is the need of the future. Again I am stressing this point because I have conducted research in we can say degrading dyes, industrial dyes, pollutants, and so on, oil spill degrading bacteria, petrol degrading bacteria, and this is required nowadays, because recently you must have encountered or read the news because of that Taukte okay, number of ships. So one oil ship, ऑयल कंटेनर लेके जा रही जो शिप थी वो कुछ सिंक हो गई, एंड बिकॉज ऑफ दैट there is large leakage of petrol and diesel. So if we want to we can say reduce it, okay, bioremediation is very, very important. Okay, so this play a very important topic. Okay, so with this I think we have covered all the topics from the applied biology. Okay, we have studied them in a brief. Okay, then हमने एक example भी देखा है. So so basically I want to tell you that focus on the topics which I have stressed. Surely it will help you, especially the agrobacterium one, because you will find one question at least in every paper. Okay, and that will surely help you in clearing the exam. So I wish you all good luck for your net set get preparation and exams. And again, I want to thank INEAS and ATBS for this opportunity. Thank you.
Thank you very much, Kaushik sir. There are a few questions.
Yes, sir.
I guess you can see in the chat box very simple questions, but as a matter they want to clear their concept.
Yes, sir.
One
yes,
question
sir.
is what is the difference between heat killed and attenuated vaccine?
Okay.
Can you elaborate on this?
Yes. In the heat killed vaccines, okay, generally, okay, just I'll start my video because I think video is not on. Yeah. Okay, so in the heat killed vaccine, we are properly, we can say, destroying the microorganism, not the structure, okay, but we are killing that microorganism either by heat treatment or either with the use of formalin. So we can use some chemicals like formalin so that the bacteria will be killed, or the microbe basically will be killed, and that will not generate any response in the host. Okay, that will not multiply. That killed bacteria will not multiply in the host. In case of attenuated, okay, we are just removing the pathogenicity. Okay, so that bacteria will multiply. Okay, it will elicit the immune response because bacteria is multiplying. So our immune cells, especially antigen presenting cells, will recognize them. Okay, it will give response to T cells. T cells will give to B cells, and that will form antibodies. Okay, so antibody production will be there, but pathogenicity will not be there. The bacteria is a very new bacteria. Okay, it useless, so it will not produce any type of disease in the host. So that is the difference between the live heat killed and live attenuated vaccine.
Question is there.
Yeah.
Live attenuated and inactivated exotoxin. The difference between both.
Live attenuated and inactivated exotoxin. Okay. When we talk about basically live attenuated exotoxin, we can say when we talk about exotoxin, we are introducing directly the toxin and not the bacteria. Okay, so generally live attenuated one, when we are introducing heat killed or just a copy of toxin, okay, that will elicit the immune response. Okay, but it will not harm the host. When we are introducing a live attenuated one, okay, maybe it can gain the pathogenicity and it can cause some harm in the individual. So attenuated exotoxin, basically a toxoid. When we talk about inactivated toxin, it is a toxoid. So toxoid is generally, we can suppress the immunity. We can suppress the toxicity, not the immunity. We can suppress the toxicity maybe number of ways, like जैसे हमने bacteria को किया था, we can use formalin and suppress the toxicity, and that will elicit the immune response. But there is a chance of we can say harm. Okay, so this is the difference.
Okay. I guess one more question from my end.
Yes.
Very basic question, but we can take it.
Yes, sir.
Can we call animal cell as protoplast?
Can we call
animal cell as a protoplast?
Protoplast.
Yeah.
Okay. Protoplast. When we talk about protoplast, the protoplasm of living plants and bacterial cells, generally protoplast are naked plant cells. Okay, so animal cell, we cannot call them as protoplast because animal cell do not consist of any cell wall at least. Okay, so protoplast are the naked plant cells. When we remove the cell wall of the plant cell with the help of number of enzymes and all, it will generate protoplast. Animal cells, we can call them as protoplast, but it's not like protoplast. Animal cells do not consist of any cell.
Okay. Sir, can you stop your slide share?
Sorry. I forgot to do that.
Jayant sir and Vikrant sir, if you have any questions from your side, you can highlight.
No.
Jayant sir? Jayant sir, are you there? Okay, fine. I guess these are all questions which we have. Thank you very much, Kaushik sir, for your elaborative talk and very interactive session. As most of the students could answer you through a live session itself. So I hereby request all the participants to join tomorrow at 9:50 exactly, so that we can start on time. And I guess you have enriched today from these two eminent speakers. And I thank you on behalf of INEAS and ATBS to both the speaker, Dr. Sudhir Verma, sir, and Professor Kaushik Inamdar, sir. Thank you very much, sir.
Thank you. Thank you.
Kale, sir, any instructions?
No, I don't think so. Not from my side.
Okay, thank you, sir. Are we stopping the live stream?